introduce today our distinguished seminar speaker, Dr. Naim Kittana, and let me to introduce him in few words. Dr. Kittana is an associate professor of pharmacology and molecular medicine at Al Najah National University, where he plays several key leadership roles. He currently serves as the director of the Medical and Health Sciences Research Center, head of the Master Pharmacology Program, and the chairman of the Institutional Review Board at the university. Dr. Kittana received his PhD in pharmacology from Göttingen University in Germany, supported by the prestigious DID scholarship for both his master and PhD studies. He was later awarded the DIDV invitation program for postdoctoral fellowship, further expanding his expertise and research experiences in Germany. With a research portfolio that spans pharmacology and molecular medicine, Dr. Kittana has authored over 30 peer-reviewed publications and currently holds an H-index of 16, reflecting the impact and relevance of his scientific contribution. We are truly fortunate to have Dr. Kittana with us today to share his insights for this presentation, and please join me in warmly welcoming him. Thank you, Dr. Kittana, for showing up on time today, and please start your presentation.
Okay, thank you very much, Dr. Ahmed, for this nice introduction. And it's my pleasure to be with you today. And it's really my honor to be invited to such an important and prestigious meeting. So I would like to thank Dr. Ahmed Salam for giving me this opportunity, and also the staff of the MGB for this nice invitation. So today I would like to introduce my work on engineered dermal tissues that is fortified by functionalized multi-wall carbon nanotubes and complexes for regenerative medical applications. But before I start with my talking about science, I would like to introduce myself more and give you an idea about my background. I'm from Palestine, specifically from Nablus city, which is located in North Palestine, a few kilometers from Jerusalem and Gaza. Actually, Nablus is one of the oldest cities in the world. Its history back for over 9,000 years. The estimated population now is like 175,000 residents in the city, but around 500,000 as a governorate. Nablus is located on and between two huge mountains, which gives it a very specific and beautiful topography and landscape. It's very modern city. As you can see here, it's very large, very huge, and very well organized, and very beautiful place. And it's famous for its iconic old city. This is really very old. This is really thousands of years ago, and it's been inhabited since ever. As you can see, there is very distinct architecture, really very wonderful. And Nablus is famous for this kind of dessert. We call it knafe Nablusiyya, and also for olive oil production, which is traditionally used for making traditional soap. And this industry is really hundreds of years ago. And interestingly, this kind of soap is being produced nowadays with this traditional method like hundreds of years ago, to give this kind of product, which is kind of, you can say it like a bio soap. It's made of pure olive oil, and so it's out of lots of chemicals. It's really very good product, and something we are really honored by. Okay, but this, of course, this nice picture I told you is distorted by the Israeli pollution, the repetitive invasion by the Israeli troops, and the tragedy that's associated with it, everyday invasions, destruction. The checkpoints are surrounding the city from all sides, so you can't move easily in and out without being endangered by these terrorist groups. Now, under this difficult situation, the Najah National University, my home university, was established in the year 1918. It's a very big university. It's the largest university in Palestine, with 25,000 students distributed across 5 campuses. Our university offers 128 undergraduate programs, 78 master programs, 13 doctoral programs that are across the 12 faculties. It's also home to 15 research centers. Our university, we are proud of it because under all these difficulties, it's according to the Time Higher Education, is positioned within 501 to 600 class globally, and for the QS ranking for the year 2025 is ranked 1001 globally and 63 on the Arab region ranking. Now let's go to science. So in my work, I'm interested in developing treatment for chronic wounds. Chronic wounds are normally extensive skin injury that takes very long time to heal, more than 14 days, sometimes weeks, months, and sometimes they never close. They can be caused by several factors like burns, pressure sores or bed sores, and also due to diseases like diabetes, which make something we call diabetic foot, that can easily develop into gangrene and may result in amputation. So there are several available pharmacological therapies for these kinds of skin injury, but the therapeutic benefits are limited. So there is still a large window for developing new novel therapies. Now, one of the materials that have shown to be promising in wound healing is chitosan, which is a natural polysaccharide, and you can use it to make a hydrogel. This hydrogel is biocompatible, non-toxic, biodegradable, and has been shown to boost collagen deposition. And you may know that collagen is the main constituent of the skin, because the skin is composed of two main layers. There's the dermis layer and the epidermis, and the dermis layer, which bears most of the mechanical properties of the skin, is made up mainly of collagen. So chitosan can boost collagen, which means that it might really help in wound healing. Plus, it has some antimicrobial activity, and then can prevent the infection, the development of infection within the wound. Now, this is something was already established by many other groups before us, but what we wanted to add at this point is to combine it with other materials. And here we thought of a material that is called carbon nanotubes. These are wrapped graphene sheets into cylinders or nanotubes like with this shape. They are present in two forms, either single wall carbon nanotube, which is composed of one layer, and it has a diameter of 0.5 to 1.5 nanometer and a length in several micrometers. And then we have also the multi-wall version, which is composed of different layers, and this can be a little bit thicker, more than 100 nanometer, but still it's very, very tiny. Now, this interesting compound or polymer has shown to have very specific characteristic that is extensively used in several industries, and we thought we could really benefit from its physical properties in our field, especially that there were some reports stating that it can cause tissue irritation and stimulate cell proliferation. So we thought maybe if we have something like that in our preparation, that maybe we can induce the cells in the wound bed to proliferate and to rapidly close the wound. In addition, carbon nanotubes are used to be toxic, but when they are coated by a hydrophilic polymer like chitosan, they become non-toxic, which is really what we wanted to have. So we started with a very simple idea that we compounded a hydrogel composed of chitosan and certain or different concentrations of carbon nanotubes, and we investigated in vitro the effect of this treatment on the production of collagen by 3T3 cells. This is a kind of cell lines, fibroblast-like cell line that deposit collagen. And as you can see in this diagram, at the y-axis we have the relative change in collagen production, and here we have the different treatments. So this is the control cell, and this is chitosan only. It boosted collagen production, and this was consistent with what we've seen before in other literature. But what was really interesting is that the incorporation of carbon nanotubes, that we have kind of a concentration dependent increase in collagen deposition, and there was clearly a difference between the two types of carbon nanotube, that the multi-wall carbon nanotube could enhance the collagen production very much higher than the single wall and by the chitosan alone. So this was kind of very promising signal for us to proceed with this project, and then from this in vitro point we moved directly to in vivo mouse wand model. And here we induced standard wands on these mice, and we treated these wands with the different concentrations of carbon nanotube within this hydrogel with chitosan. And as you can see here, with the presence of, or with this treatment, there was better wand closure that could be observed by both the single wall carbon nanotube and the multi-wall carbon nanotube. We also performed histological evaluation for these tissues, and we found that with the presence of the multi-wall carbon nanotube, there was more fibrosis. There was more collagen deposition. So this can be a signal for enhanced wand healing. In addition, this is also was very important, that there was a positive effect on the thickness of the epidermis layer. So the epidermis was increased by most of the treatment. So from that we concluded that we really have a topical treatment that could have promising effects in the treatment of wands. And these data were published in the Journal of International Nanomedicine with impact factor 7.5. Now we wanted to take a further step within or based on our findings, and we decided to move to the engineered dermis tissues. So we wanted to generate engineered dermis tissues that consist of collagen and chitosan, so that it will be incorporated within the wand bed, and hopefully that we get a better outcome. And one of the things that made us think to go or to utilize our product in engineered tissues is that the current engineered dermis tissues suffer from weak mechanical properties. So there are already several research groups that generated engineered dermis tissues based on collagen, but these tissues were very fragile, and when they were implanted in one bed, they tended to disintegrate, to break easily. They are very difficult to handle, and since carbon nanotubes have distinct physical characteristics, they have elasticity which is greater than steel. As I showed you here, it has a strength of 50 times greater than steel. This is very tremendous, you know. So we thought we could utilize this, and we thought if we could incorporate this carbon nanotube within the engineered tissue, we might increase the strength of this tissue. And for that, let me go back to where we ended. We thought that we would then generate this engineered tissue utilizing chitosan, which is a kind of sugar, biodegradable polymer, so it will go with time. It can induce collagen, and it will be combined with collagen. So our tissue the matrix of our tissue will be composed of collagen, chitosan, and the multi-wall carbon nanotube. And before we go to check its effect or potential effect for the treatment of chronic wands, we investigated its mechanical properties. So here we generated engineered dermis tissues that composed with collagen, multiwall carbon nanotube with chitosan, C stands for chitosan, and this tissue contained human foreskin fibroblasts. So these are primary human fibroblasts. This tissue was casted in this plate that contains these kind of strictures, as you can see here, and the tissue was casted in the form of a ring. After like 7 days, the tissue condensed, and the cells within the tissue started to contract the matrix, and the tissue was very consistent and very well integrated together. So now we tested the mechanical properties of these tissues. So we fixed this tissue in a rheometer device that pulled the tissue apart, and while pulling it measured the stress and the strain. The stress is the force per cross-sectional area that was used in the pulling process, and the strain is the relative change in the tissue size in the extension. So this is dimensionless because this is kind of proportional change, proportional deformity of the tissue. As the apparatus started to pull the tissue, the stress was increased with the strain, initially in a linear manner, and within this region, then since it's linear, then this is kind of an elastic extension. Elastic means that if we release the tissue, it will come back to its original shape and size, which means that during this pulling steps, the collagen fibers are still intact, they are not disrupted, they are not broken, the tissue is not harmed. Now, at this point, the behavior of the tissue started to change. It's no longer linear, and this is what we call the plastic strain. The plastic strain, which means that the tissue is extended, but if released, it cannot come back to its original shape and size. And the reason behind that is that the collagen fibers are started to rupture, to break down, and they cannot then recoil again to the original shape. So then when we continue this, the slope started to decrease gradually until we reach this point we call failure point, where the tissue ruptured. It was broken at this point. So from this graph, we measured several important mechanical properties. First, the slope of the linear range, and this slope is called the Young's modulus, and this represents the stiffness. The higher, the more slope, then you have stiffer tissue. Also, we have this yield point. This yield point, this is the point where the tissue started to change its behavior from the elastic behavior to the plastic behavior. So this point is determined by what we call the stress, the yield stress, and the yield strain point. Also, at this point, this is the maximum strength, which is the maximum stress that is measured that induced by this tissue before it started to break, to physically break. So this is what we call the strength of the tissue, and this is what which is the maximum stress point. Also, we look at the area under the curve. This dotted region, this is what we call the resilience of the tissue, which is the area under the elastic region of the curve, and the area here represents the tissue toughness. So these are several mechanical properties that we could learn from this experiment, and this is what we had. So initially, these are the tissues that we generated. These are the control tissues, which is tissues without chitosan, without carbon nanotubes. It's made only of cells and collagen. And here we have with collagen and with different concentrations of multiwall carbon nanotubes, and these are representative images for the stress-strain curves. As you can see, there are really very big differences. So if we look at details rapidly, we can see that With the multi-wall carbon nanotube, we have kind of increase in tissue stiffness, but only with this concentration of the carbon nanotube. The yield stress, or what we call the proportional limit, was increased by the multi-wall carbon nanotube. Also, the elasticity increased, the resilience increased, the strength increased, the ductility was increased, the extensibility, which is how much the tissue was extended before it break, and also the toughness. All these parameters were increased in the presence of multi-wall carbon tube, though the tissue has become more elastic, is a little bit stiffer, has greater resilience, greater extensibility, greater toughness, which means can endure more physical stressors. And this is actually what we really wanted to have. We made histological analysis for the tissues by using tritic phalloidine stain to stain the cytoskeleton of the cells, and also DABI to stain the nuclei. And we've seen that the cells were integrated within the tissue. They were viable. They did not die due to the presence of carbon nanotube. And the cells were organized in lines across along the lengths of the tissue, which means that the cells are functional. So they are there, they are functional, they are viable, and the tissue's mechanical properties are really enhanced. Now, these interesting results were also published in the International Journal of Nanomedicine. Now, to go further, now to check whether these tissues now can improve the healing of wounds, and therefore now we went to in vivo. And here we wanted to investigate the healing properties of these tissues. But here at this point, we thought of also incorporating a growth factor like angiotensin II, because angiotensin II has been shown to be involved in wound healing, not only in cardiovascular diseases and the regulation of blood pressure, but it's also involved in the behavior of fibroblasts in the skin. So we thought maybe adding angiotensin II could also be beneficial. Now here we generated in new tissues, but this time we generated them in the form of patches to be suitable for transplantation into wounds. And then we went to mouse model. One mouse model, we created identical wounds on either side. So one side will receive the treatment tissue, and the other side will be the internal control for this for the treatment side. So each mouse has its own internal control, so that we can minimize the individual variations. So the tissues then was wrapped with tegaderm, this kind of polymer that is used clinically to protect the wounds. This was, of course, very important to keep the tissue in place and to prevent its dryness. Now, after 14 days, the animals were sacrificed, but before sacrificing them, we imaged the mice at day 0 and at day 14, which is the day of euthanization. So we have here the upper panel. This is for day 0, and the lower panel for the same mice after 14 days. So we have here the control angiotensin II group 0.025 with angiotensin II, 0.1 and 0.1 carbon nanotube with angiotensin II. We made macroscopic analysis for the wound closure, and these are the parameters that we used. So here the dotted red line represents the initial wound area. Then we have this blue line. This represents the non-contracted area, and here this is the contracted area, because this one is not healing. So the one started here and then it ended here, but this is a normal skin. This is healthy skin. So why it's brought here? Because during wound healing there is a contraction process. So the wound normally, this is the normal phenomenon, that the wound contract to minimize the size of the wound. So this is the contracted area, and this is the non-contracted area. Also, we can see here, this is a newly healed area. So this is kind of what we call epithelialized area. And this is what we have in the center is the open area, which is what remained from the wound and still open, is not healed. So from these main regions, we could calculate the percentage of the non-contracted area, which was calculated by dividing non-contracted area by the initial area, then the percentage of the epithelialization. Then we have, and for that we have the epithelialized area divided by the initial area, and the wound, or the percentage of wound closure, which is the closed area divided by the initial area. And what we had was really interesting. For the non-contracted area, it was all increased. So what does it mean? Normally, when a wound heals with contraction, this causes distortion of the skin texture and appearance, and it will leave scars at the place of the wound and marks that will stay forever at the site of injury. So wound contraction on one side is useful to accelerate the wound healing, but on the other side, it can disfigure the shape of the skin. So since we reduced the wound contraction, this was what we consider a positive effect, that this will reduce the scarring of the tissue, and the tissue will heal with better outcomes. Also, the epithelialized area was increased, but here it was increased more or significantly in the presence of angiotensin II. This is all we were lucky that we added angiotensin II in our study. Otherwise, we could have missed this promising effect. Now, angiotensin II, we've shown that it increases the epithelialization. It increases the growth of the epithelial tissue, which is a very crucial step in wound healing. However, the wound closure at the end was not changed, let's say. The histological analysis for this transplanted tissue showed that there was no change in the thickness of the epidermis or the thickness of the dermis. This is good because we don't want it to be changed. We want it to be normal. We don't want to have a lump of tissue under the healing wound. So the tissue actually resolved within the tissue. It integrated very well, and the excess tissue maybe was resolved somewhere. So the histology was in general normal. So in conclusion, of course, this work is submitted to a journal recently, and we hope to have good news very soon. So in conclusion, the engineered dermis tissue containing chitosan and multiwall carbon nanotube have improved the biomechanical properties of the tissues, or the EDTs can improve the quality of wound healing in terms that it enhances wound epithelialization and reduce wound contraction. And at the end, I would like to thank my group and colleagues in our university, and I would like to thank you also very much for your attendance and hearing.
Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow.